afternoon and welcome to our Food Safety and Science Across Canada webinar series. My name is Gwendolyn Jones and I have the pleasure of being the Executive Director here at the Institute for the Advancement of Food and Nutrition Sciences. For those of you who are not familiar with us, want to emphasize a bit on who we are. We mobilize our membership from government, industry, and academia to drive, fund, and lead actionable science, focusing on food safety and nutritional science, all dedicated to advancing public health. We catalyze the creation of scientific knowledge by funding independent research, and we also convene experts to drive positive change in the food and beverage ecosystem. Our collaborative and inclusive structure empowers our members and our community to bring forward the diverse perspectives that we all know and welcome. Today, we're going to hear from three esteemed researchers in this webinar. We're going to hear from Dr. Tom Gill on bioactive peptides. Then we're going to hear from Dr. Rotimi Aluka on anti-hypertensive peptides. And then we're going to hear from Dr. Johnny Rollin on the identification of low molecular weight peptides from food proteins. So we're going to have a really interesting series of talks that really walk through food innovation and how it can in fact move from, research in a laboratory towards commercialization, right? Towards having impact, because as a science organization, and dare I say, all of us as scientists, we like to know that the work that we do is highly translatable. We're meeting today on a GoToWebinar platform, okay? And within the GoToWebinar platform, I want to highlight a couple of things. The first is that we've got a button, and you should see it on your screen on the right-hand side up at the top. That is the control panel. With that button, you can expand or collapse the size of your control panel. Additionally, want to highlight, and this is actually very important, that we want to make this a little bit interactive, which means that we want you to bring forward questions that you have for our presenters today. If you go ahead and type in questions or comments within that question box, after each of the speakers, we'll be able to ask them questions. Please note that the questions that you ask are only visible to IFIN staff, right? They're not going to be visible to other attendees, but we on the staff side will be seeing them, and we will bring forward your questions to the presenters. So without further ado, I'd like to invite Tom Gill to join me on the screen, and we will also send over to Tom the presenting rights. So Tom, you want to click on your camera button and your microphone. Excellent. There you are, sir. Good to see you.
Okay, great.
And looks like we've made you the presenter. If you want to go ahead and share your screen.
Okay.
We got it. The floor is yours, sir.
All right. Well, I'm going to kick things off just by giving some of the background on how this work that we've been involved in began. And I think it's a good example of basically government-funded research at the universities and moving into sort of commercial production. This isn't the first time I've been involved in projects which involved industry, but way back in 2007, I was involved in a network of centers of excellence called AFMnet. And during that time, I met a lot of people from different disciplines, and I think that was the whole gist behind setting up these networks of centers of excellence was to get people with different backgrounds working together. So we had a conference in 2007 out in Vancouver, and I heard a gentleman at the time from Laval University, a nutritionist, talking about seafood proteins and about his work on type 2 diabetes, and his name is Dr. Andre Maret. And I met Andre for the first time right after his talk. He gave an inspiring talk, and it got me thinking about possibilities. He talked about possibilities of using dietary protein to control or mitigate type 2 diabetes. And so that's how things started out. I invited him out to dinner that night, and a bunch of us went to a restaurant, and I sat beside him, and we shared a bottle of wine. And I said, Andre, the work that you've been doing on fish proteins really excites me. And I'm just wondering, so basically, I'm going to move into the slides. The work you've been doing on type 2 diabetes is extremely interesting. And the fact that proteins could manage to mitigate, I said, I really wonder whether it's proteins or whether we're talking about fragments of proteins, peptides, because after all, when we ingest a meal of proteins are broken down by digestive enzymes and chemicals in our body. And the bottom line is that in order for the proteins to be absorbed through the intestinal mucosa into the bloodstream to be effective, they've got to reach a certain sort of maximum size, and only maybe 2 or 3 or 4 amino acid containing peptides. And so I'm interested in looking at peptides, and I have been working on peptides for a long time. So we decided that I would write a proposal to NSERC, the Natural Sciences and Engineering Research Council of Canada, which I did the following year, and managed to get $500,000 to look at the idea of taking fish proteins and digesting them in vitro in the lab with the same enzymes and same chemical conditions, and seeing what kind of peptides we would generate and whether any of them would have anti-diabetic activity. And so this was our hypothesis, that it wasn't the proteins per se, but it was the peptides, the very low molecular weight peptides. And so that was the starting point. And it was basically, I had never thought about it before that day, and that's sort of how it all started. So we got the money, and I started to investigate about type 2 diabetes. I'm not a nutritionist, I'm a food scientist. And so the deal with type 2 diabetes is that in early stages, patients produce normal or even elevated levels of insulin, unlike type 1 diabetes. So these people, generally speaking, aren't short on insulin. Insulin is the hormone that the body produces in the pancreas that allows the incorporation of glucose, dietary glucose, from a meal that you eat to be mobilized, go through the intestinal mucosa, and get internalized in the cells. And roughly 80% of the energy produced from the glucose in the blood, it's produced in the myofibrils, so it's induced in the muscle cells per se. So the muscle uses this energy, and the problem with type 2 diabetes is that the action of insulin becomes blunted. And by blunting, I mean given a normal amount of insulin, your body doesn't react the same as it does in a normal individual. It results in elevated blood glucose, and if left untreated, can lead to fatigue, blurred vision, thirst, slow healing sores, frequent infections, cardiovascular disease, a number of other things. So anyway, this is how it all started. Now, I'm not an expert, I'm not a physiologist, but there's several things that go on here. You can see at the bottom of the screen the muscle. So the muscle needs glucose, and it's the insulin that results in the internalization rather. Keep getting a slide. There we go. Internalization of glucose into the muscle so that we have muscle contraction, and the energy is generated through ATP through the general pathways. Anyway, also as part of type 2 diabetes, the liver tends to produce more glucose than normal. So you've got a flooding of glucose. And at the same time in the gut, we have an impairment of the incretin effect. There are hormones in the gut which result in the production of insulin. So when you have a big meal that contains glucose, the gut automatically begins to produce more, or at least it signals the production of insulin, and the insulin is produced in the pancreas. And at the same time, during type 2 diabetes, we have an increase in the rate of cell death and a decrease in the beta cell mass, the cells that produce insulin. Concurrent to all this, we have a change in the adipocytes, the fat-containing cells. The circulating fatty acids within the bloodstream levels increases, and we get a condition called hyperlipidemia. So that's sort of some of the things on the background. So a lot of things can happen to you with type 2 diabetes, but it's all related to the impairment of insulin. I'm going to say a few words about bioactive peptides. I've been working on them for most of my career. Bioactive peptides are ubiquitous in nature, animals, insects, and bacteria. Early studies have shown that dietary fish protein has a positive effect on the markers for type 2 diabetes. And the conference that I was at, that I heard Andre Maret speaking at, talked about a study, an earlier study that had been done about a year before, and showed the marked improvement in insulin sensitivity in human subjects that had been fed under clinical trials a cod protein diet for approximately 4 weeks, compared to patients that had been fed diets containing lean beef protein, pork protein, veal protein, egg protein, milk and milk product proteins. So it turned out that cod, in this case, seemed to improve the condition of type 2 diabetes, at least in human subjects, which is exactly what we were looking for. So this gave us a hint as to what was possibly going on. And so the hypothesis that Andre and I drew up was that it was the hydrolyzed fish proteins. So I decided to go to the Natural Sciences and Engineering Research Council for money. And back in those days, you had to have an industrial partner in order to get a grant. So I was looking for relatively big bucks, about $500,000. And at that time, there in Canada, there was only one company that was producing um, lean fish such as cod and salmon in captivity. And I thought this was essential because when you're dealing with fish protein, you need to get it really fresh. So farmed fish was was what we were looking for. And so the way I wrote the proposal for the research was, okay, so there are two upsides to this idea. We're going to use fish waste, protein from fish, which is normally thrown away or turned into fertilizer or feed. We're going to use that leftover protein, and we're going to come up with peptides that reverse or at least modulate the effects of type 2 diabetes. And so that was the idea that I sold the Natural Science and Research Council on at that time. So we decided that we were going to do that, and I came up with a process, which I'll discuss in a minute here, on how we actually did it in vitro in the lab using the three enzymes, pepsin, trypsin, and chymotrypsin. But in the meantime, I'll talk about how we screen. So you know what you're going to do, and you come up with a fraction of peptides, and you have to come up with a rapid enough method for the analysis, whether these peptides that you've isolated and so there were three tests that efficacious or not. Andre set up. One was so the idea is you grow these cells up in cell culture in petri dishes, and you then feed the radioactive glucose and either add your peptide or not add your peptide, and add your insulin or not add your insulin, and then test for glucose uptake. And if you get an increase in glucose uptake into these cells, these muscle cells, you consider that a positive effect. Okay. There are two other tests that they used, and one involved hepatic cells, and the cells, as I said, in the liver, and these were again rodent cells. They're produced the insulin. In the case of type 2 diabetes, you get an increase in the amount of sugar, glucose produced in the liver. And a third one involves the adipocyte. So we're going to get to that in a few minutes. Anyway, so we started out, these are salmon frames. We started working on salmon because quite shortly after we began the project, Cook realized that it was going to be too difficult to grow codfish in captivity, and they reverted back to salmon. So you wind up filleting the salmon, and after you fillet them, you get these frames, and in the center of the slide, you can see it's basically salmon hamburger. It goes through a machine and it extracts the myofibrillar protein from extremely fresh material. This is typically only a day old or less. And eventually you digest it, you fractionate it, and you come up with something that looks like, on the right-hand side, ginger ale. Or at least it looks like ginger ale, doesn't taste like ginger ale. But you get solubilized peptides, and most of them, 99.9% of them, have molecular weights of less than 1,000 Daltons. So the process involves, first of all, we take that fish hamburger in the middle of the slide, we dissolve it in 1 normal sodium hydroxide and dissolve the proteins, make them all soluble, and then we isoelectrically precipitate them with hydrochloric acid to give us an isoelectric precipitate at pH 4.5. And we collect that precipitate, and then we digest in first with pepsin, at a low pH, and then with a mixture of trypsin and chymotrypsin at a higher pH. We've got a solubilized protein now, something that's very, very soluble and low in molecular weight. And then we filter it, and we furthermore ultra filter it until we get a fraction that's less than 1,000 Daltons. And because we've used sodium hydroxide and hydrochloric acid, we've got a lot of salt in that ginger ale, and we've got to get rid of that. And we use nano filtration, well, there are several ways of doing it, but basically using a filter with a membrane filtered with a very, very low molecular weight cutoff. And you can successfully remove the salt and the water and concentrate the peptides down using this finished step, and then we freeze dry. So this is a picture of the mechanical deboner. Here's a whole fillet going into the mechanical deboner. Normally we use frame material, and when we're all done, we throw everything into a tank, and this isn't how we started out. We started out with beakers. This is a commercial, well, semi-commercial scale digestion tank. I think it's 1,000-gallon tank, and we've gone way, way up now, and we're producing it in large batches. This flow chart just shows the various steps, and you can go through it in detail. It gives all the details of how we did it. I've had several students work on this project, on various aspects of this project, and Johnny Rowland, actually, who's going to be presenting in a few minutes, was my last and final grad student who finished his PhD a couple of years ago, and his job in his PhD thesis was to identify the amino acid sequences on the bioactive peptides that were of interest in controlling type 2 diabetes, and he'll talk more about that. And the work that he did is now been patented. Johnny and I hold the patent, and it was patented through Cook Aquaculture, who are our benefactors and are planning to go into commercial production. I probably put this in the wrong order. This is about glucosynthesis in the liver. That was test 2. And test 3, looking for activity, involved measuring nitrous oxide production in macrophage. And I'm not going to go into this because when Johnny did his thesis, these tests are very, very slow going. They take a lot of work and a lot of money to produce. So we wound up just screening using glucose uptake into cell culture muscle cells. So Johnny is going to talk about how we identified the various components of the bioactive material. As I said, the patent's been applied for, so we can sort of give you some details on it now. And before I go too much further, I'm going to finish this off by talking about, so you're into production, at least not commercial production, but pilot scale production. We had to find a way, after we had identified the active ingredients, we had to find a yardstick to measure how much of these peptides were produced at the end of the day. So it takes about a week. It takes a 5-day week to produce a batch of these peptides. So at the end of the week, you have to know whether you were successful or whether you weren't successful in producing these bioactive components. And since then, the work I've been focusing on, this hasn't involved Johnny, but it's stuff that I did on my own through the company. And we've been developing and have developed now an immunoassay which identifies the 7 peptides individually and in combination that are produced and that are bioactive. So that's kind of what I'm doing. I'm keeping myself out of trouble. I am retired, but not really. I'm still busy working on this. Now, during the process, I ran into a fellow by the name of Rotimi Aluko, who's going to be our next speaker, I think. And Rotimi and I got working on some work we had done looking for other bioactivities in the same idea. And we actually produced bioactive peptides from cod and salmon, took them out to Rotimi's lab at the University of Manitoba, and he's going to talk to you more about that. So that's about all I have to say for now, and I'd be more than happy to answer any questions you have. Hello.
Great, thanks, Tom. Appreciate your giving us that overview on how you came to do some of this exciting and highly translatable work on biopeptides, particularly coming out of salmon, and also appreciate your nicely setting up both Dr. Uluko and Dr. Rollin, who we'll hear from later on today. As we think about questions that folks have coming in, and I've got a couple. The first one is, does the peptide that you were talking about have any effects in lowering blood pressure?
I'm going to refer that because that's part of Dr. Aluko's talk. So if you'll just wait for a few minutes until he presents the data, because he worked on basically the same peptide preparation that I worked on. I made the peptides and gave them to him, and that was one of the bioactivities that he looked at. So that's coming up.
Excellent, excellent. Then could I ask you to reflect on how you see differences in how research is, dare I say, done and pursued between what you were initially looking at as an academic researcher, right, and where Koch Industries and others are looking to go with the same type of science? I know a lot of our audience includes graduate students and others who are looking to, I'll say, launch innovations in that space. Could you just kind of just provide a few remarks on that?
Sure. Well, I actually talked to Rick Yatta today, who was the executive director of AFMNet, to see if he could remember when we had our meeting in Vancouver, and I think it was 2007. We agreed on that, but at any rate, it was a eureka moment. If you've ever been in an audience and listening to somebody speak and they stimulate you so much that the hair rises on the back of your neck, you know you've got something that's worthwhile. And that's exactly what I thought. As soon as he started to talk about these proteins, I had an idea, and it just came to me. It was pure serendipity, but that was like more than 10 years ago. And we stuck with it, and it will be a commercial product, if not a drug on the Canadian market at some point in time. But it just seemed to fit. I had a lot of collaboration with seafood processors from all over the world in my earlier days, and as soon as he started to talk about salmon and cod, I thought, well, I know who our commercial partner could be, because they were the biggest in Canada and the only company that were at that time producing aquaculture, farmed cod and farmed salmon. I know there's been a lot of bad publicity on fish farming, but in this case, if you're manufacturing a drug, or initially it will be a nutraceutical, it was a natural selection for me, and everything just seemed to flow. And all I can say is be persistent. If you know you're right, go ahead and do it until it's done.
Excellent. Thank you very much, sir.